Next up, our final presenters of the panel, we have a team, Tara Chen and Meera Chopra. Tara is entering her third year of the Bachelor of Medical Sciences program at Western University. She has completed several computer-based projects, including the development of a skin cancer classifier based on artificial intelligence and the creation of mathematical models for a synthetic biology project. In her spare time, Tara enjoys watching nature documentaries, playing piano, and hiking. Meera, on the other hand, is entering her third year of health sciences at McMaster University. She's interested in how genetics and the environment can influence our health throughout our lives. She's worked on multiple genetics-related projects in the past, including a systematic review on the epidemiology of genetic disorder causing tumor growth in the nervous system. In her free time, Meera enjoys reading and cooking. Here they both are on Inhibition of MALT1 by Mepazine Acetate for the treatment of psoriasis induced by CARD14 mutations.
Hi everyone, I'm Tara and I'm from Western.
And I'm Meera and I'm from McMaster.
And we're studying in the COBWEB program at the University of Toronto under the supervision of Dr. Brad Bass. Our project is on the effectiveness of a novel psoriasis treatment. Psoriasis is a chronic inflammatory skin disease characterized by red and scaly patches on the epidermis, and it is a result of overactive immune responses. Psoriasis can appear at any age. However, genetic and environmental factors affect individual susceptibility and presentation. Certain alleles located in the major histocompatibility complex can cause a 60 to 90% chance of heritability of the disease. As well, environmental triggers such as UV exposure and smoking may cause psoriasis to appear in genetically predisposed individuals. One cause is attributed to genetic mutations in the CARD14 protein, which causes overactivity of the NF-kappa B pathway. Specifically, the overactivity of the pathway is caused by an increase in the activity of the protein BCL10 and the protease MALT1. This CARD14 BCL10 MALT1 complex, also known as the CBM complex, activates NF-kappa B signaling, which increases pro-inflammatory gene expression. As a result, keratinocytes in the epidermis are overproduced and accumulate, creating psoriatic plaques on the skin surface. One potential therapeutic compound is Mepazine Acetate, which allosterically inhibits MALT1. Currently, psoriasis treatments use topical therapies. Topical corticosteroids are commonly used for mild to moderate cases of psoriasis. They upregulate the transcription of genes with anti-inflammatory functions, including the IL-1 receptor antagonist, IL-10, and tyrosine aminotransferase. However, patients who receive topical therapy often report minimal symptom improvement. Another therapy for more proliferative and severe cases of psoriasis is ultraviolet phototherapy. Research suggests that ultraviolet light causes immunosuppression due to the induction of apoptosis in both keratinocytes and T cells, which are involved in pathways causing psoriasis. Current therapies that are being developed include the use of biologic agents for the treatment of psoriasis, which, for example, compete for the binding site of TNF-alpha on TNF receptors 1 and 2. The actions of TNF-alpha typically activate the proliferation of macrophages and stimulate the release of interferon beta, resulting in the characteristic inflammation of psoriatic plaques. However, topical treatments do not significantly alleviate symptoms for severe cases. We propose an alternative method of drug delivery with a transdermal patch to treat severe cases of psoriasis. Transdermal patches provide a specific dose of medicine, which travels to the stratum basale of the epidermis, where the drug interacts with MALT1. The objective of our study was to determine the therapeutic impact of MALT1 protease inhibitors on the proliferation of psoriatic plaques using a computer simulation.
So the computer simulation that we used for our project is called COBWEB, and this is used to model biological system. Agents, which are the primary decision makers in cobweb models, are represented by triangles with a colored dot in the center, and different colors are used to differentiate between agent types. Their primary sources of energy are solid colored squares named resources. And based on different parameters set by researchers to create unique environments, agents are able to make different decisions regarding moving, collecting resources, and breeding. We use ticks to measure arbitrary units of time in our model. And you can see our cobweb model is split into 3 layers with 1 cell type in each layer. So we use the Agent Abiotic Factors tab to split the model, which is a 100 by 100 grid, into 3 layers. So the bottom section represents the stratum basale of the epidermis, the middle is the stratum spinosum, granulosum, and lucidum layers, and the top represents the stratum corneum. Cells that are activated to produce CBM complexes are labeled as diseased and are represented as a navy blue triangle. And they are able to pass on the disease through contact with other skin cell agents, which is how we represented genetic mutations causing psoriasis. So the blue agents at the bottom, representing keratinocytes, produce CBM complexes after the activation of the dectin-1 GPCR by interferons and Th17 cytokines. These are confined to the lower layer of the model. And the yellow agents represent polygonal keratinocytes, and they can be induced with the disease through contact with diseased blue agents to produce CBM complexes as well. These agents are confined to the middle layer of the model. And the green agents represent dead keratinocytes present in the stratum corneum. And after contact with the diseased yellow agents, these agents age at a quarter of the rate of their healthy counterparts, resulting in a buildup of these dead cells on top of the skin. And this buildup characterizes psoriatic plaques. And these agents are confined to the upper layer of the model. So all 3 of these agents are set with an age limit to represent the lifespan of keratinocytes. And agents are initially present in small quantities and allowed to proliferate until 2,500 ticks. At this point, the model of the skin is fully developed, and you can see plaques at the top layer. So in order to closely mimic psoriatic plaques in the stratum corneum, we turned on the swarming function of dead keratinocytes at the top layer. And agents are able to group together, demonstrating inflamed areas that are characteristic of psoriasis. So the red agents in our model represent the Mepazine Acetate treatment and are initially placed in the bottom layer of the model. These agents represent an allosteric inhibitor of MALT1 present in CBM complexes. After being placed in the lower layer of the model initially, they're able to move freely within the grid. And after they come into contact with diseased agents, the diseased agents are healed and do not further propagate the disease. For a control simulation, agents are allowed to proliferate for 37,500 ticks. And for experimental simulations, we use the same parameters as the control. However, at 2,500 ticks, treatment agents are inserted into the lower layer of the model to represent the systemic delivery of the treatment. And the model is allowed to run for 35,000 more ticks, representing a full week of treatment. We conducted 25 control and 25 experimental trials.
Discovered that the amount of CBM complex producing dead keratinocytes in the treatment trial decreased over time with a slope of negative 0.0018, but increased in the control trial with a slope of 0.0002. The mean total number of CBM complex producing dead keratinocytes after 35,000 ticks was also measured, and it was determined that the number of CBM complex producing dead keratinocytes in treatment trials was significantly lower than in the control trials. On average, control trials shown in blue had an average of 662 cells contributing to plaque buildup, whereas treatment trials shown in red had an average of 620 cells, which is significantly different. Overall, the transdermal patch delivering the Mepazine Acetate treatment was able to reduce the number of psoriatic plaques on the epidermis and decrease the proliferation of psoriasis.
So, due to the systemic drug delivery mechanism of transdermal patches, mepazine acetate may be used more effectively to treat moderate to severe psoriasis for patients who have not found success with topical treatments. The therapeutic drug travels through the skin and disperses throughout the body via the circulatory system. There is currently a patent for the usage of the S enantiomer of mepazine acetate to inhibit MALT1 to target diseases, including cancer and autoimmune conditions. In a 2014 study, mepazine acetate was used to treat a murine model with multiple sclerosis. This treatment reduced the response of peripheral autoantigen-specific T-cell responses without affecting regulatory T-cell development. This demonstrates that mepazine acetate has the therapeutic potential and is able to reduce harmful T-cell responses without affecting T-cell responses that may be necessary to protect ourselves from environmental threats. A limitation of our study is that the model that we used is an idealistic representation of psoriasis, since we used a simulation. Since clinical cases don't present as ideally as software models, there will obviously be variation between our results and clinical findings. So future directions of our study include using more complex modeling software in order to represent immune mediators and individually stratify layers of the skin so that it's more similar to clinical cases. And we would like to acknowledge everybody in the University Research Experience with Complex Systems program at the University of Toronto, especially Dr. Brad Bass, and we thank him for his support and guidance throughout this entire process. Thank you for listening, and feel free to ask us any questions that you have.
Thank you very much. That was an excellent presentation. So we will indeed open us up to questions here. I do have one for you too, and maybe I just missed it when you were covering it. But you said that typically what's been used in the past for psoriasis as a therapeutic intervention has been topical steroid therapy. And do you know why, or do you have any reasons as to why it's speculative that it doesn't seem to work in a lot of cases and that a lot of people are unsatisfied with it?
Well, it does work for the more mild cases, but for moderate to severe cases, it hasn't really shown much improvement for those patients. In the paper that we read, I don't recall that they said anything in particular as to why it didn't work for the severe cases.
Yeah, Tara covered really well the paper that we read about topical treatments in psoriasis. But this is just speculation, but I've heard some cases of patients' skin becoming resistant to the treatment. So although corticosteroids are going to work initially to treat the psoriasis and kind of reduce a lot of the inflammation, eventually our skin just becomes like used to it, and the inflammation and red and scaly patches happen again. Which is really unfortunate, but definitely another future area of study within dermatology.
So I would, if I can, I'd like to follow up with another one. Is the patch that you guys proposed using, is that immune to the sort of tolerance problems that the topical steroids sometimes have?
That's a great question. Our study, the one that we used, the model that we use right now, just represents like the initial treatment. So like the first time you slap on the patch, see what happens. It would be really interesting to see if there's any tolerance. Like if we run our model for longer, if the patches appear again.
Yeah, for sure. That'd be a great thing to put in the next study. Thank you.
This might be kind of a layman's question, but is psoriasis similar to, related to, or a different form of things like eczema as well?
Yeah, that's a great question, actually. Yeah, they all kind of have like the way that they present in patients, and they all also are treated by corticosteroids sometimes. So a lot more research needs to be done as to what actually causes all of these conditions, but the way that they're treated are pretty the same.
I've got another. So you mentioned that these patches seem to be gaining some momentum. You've got a patent. You're starting to run more and more complex models to see how it'll react with psoriasis. But I'm wondering if there have been, and if not, how far along are we till we're doing like proper clinical trials and really considering this as a go-to kind of therapy, maybe in place of the topical steroids?
Well, patches have been used to, with MALT1 inhibitors, they have been used in patches to treat other similar diseases. But as for psoriasis, there hasn't been any yet. I mean, currently, me and Meera obviously don't have the financial funds to actually do clinical studies. So hopefully, maybe our research would motivate institutions to consider this.
Might make a good topic of study for a master's program.
Yes, definitely.